Hi again, my name is Megan Leach and today is June 22nd, 2021. So today we're going to be talking about the determination of gender, specifically looking at the different genes and hormones and how that will influence what gender an individual will be. So the sex chromosomes are what determine the genetic sex of an individual. They are inherited from the parents as seen right here in this image, and males have an XY chromosome and females have two X chromosomes. So the mother will always inherit an X chromosome and the father will either inherit an X chromosome or a Y chromosome. This means that the Y chromosome is what is the deciding factor if someone is a male or female. So the father will inherit the Y, and then the mother will inherit an X, and that will result in a son. If the mother inherits an X and the father inherits an X, he will result in a daughter or a female. So the sexual differentiation occurs early in the development. So prior to differentiation, embryonic tissues are considered bipotential. They're considered bipotential because it's too early in the development to determine which sex they are. A bipotential will consist of a Wolfram duct and a Mullern duct, as we see right here and right here. So both of these have, both sexes have both ducts at this time. And then the sex determination will depend on the presence of the SRY gene. The SRY gene is what determines what the gonads will develop into, what ducts will proceed, and what ducts will no longer be there. Males have the gene, and females lack the gene. In males, the gonads will turn into testes, and in females, the gonads will turn into ovaries. And this solely is based upon the fact of the SR gene being present or not. Once again, in the males, it is present, and in females, it's not present. So let's look specifically at the male embryonic development. So as we stated earlier, the male embryonic development, it consists of an XY chromosomes, and it has the SRY gene present. So the SRY gene will produce a protein that binds to DNA and activates additional genes. These additional genes consist of SOX9, WT1, and SF1. These three genes will have a direct development of the gonadal medulla and the testes. Once the testes have differentiated, they will then secrete different hormones. These hormones consist of glycoprotein anti-Mullern hormone, also known as AMH, and then also has androgens, and that those consist of testosterone and DHT. DHT is the derivative of testosterone. So the AMH hormone will cause the embryonic Mullern ducts to regress, meaning they will no longer be within the male embryo. Testosterone, on the other hand, will convert the Wolfram ducts into the male accessory structures. These accessory structures consist of the vas deferens, the epidermis, and the seminal vesicle. Testosterone will then later control the migration of the testes and from the abdomen to the scrotum, and the remaining sex characteristics of the male is controlled by DHT. The DHT, since it is the derivative of testosterone, it binds on the same androgen receptor as testosterone, but as stated, elicits a different response. So testosterone solely acts on the Wolfram ducts, and DHT acts on the different I just drew a blank on the different remaining sex characteristics. So in these two images, it's kind of a summary of what I just said. So here is right after you have the bipotential embryo. So that would be right here normally, and it comes down, and you have this male embryo at 10 weeks. So as seen here in this image, the SRY gene in the male embryo will direct the medulla of the bipotential gonad to develop into testes, right here. And the anti-Mullerian hormone from the testes causes the Mullerian duct to disappear. So in this image, they are no longer here, as we will see them in the female embryo. And then at birth, the testosterone will convert the Wolfram ducts in the seminal vesicle, which is right here, the vas deferens here, and the epididymis right here. DHT will then go on to control prostate development and then also all other sexual characteristics. Right here is just another quick summary saying that the sex determination is based upon the Y chromosome and then also the SRY gene, which then produces some other proteins then go on to have influence on the development of the Wolfram duct and then regression of the Mullerian duct. And here we can take note of where these two hormones are secreted from. So the AMH hormone is secreted from serotol cells and the testosterone is secreted from interstitial cells. And then when we go on to look at the female embryonic development, we can take note from earlier slides that it is in 2 X chromosomes. There is no Y chromosome involved, and the SRY gene is not present. So when we look back at our bipotential embryo, the gonads in the female will develop into the ovarian tissues. So in the text, it noted that there's a large number of female-specific genes that also influence the development into the ovarian tissue, but they're not very well known, and so it took note just to mention that there are other genes at play here besides. There are other genes here at play. Sorry. With no testicular AMH, the Mullerian ducts will then develop into the upper portion of the vagina, the uterus, and the fallopian tubes. There also is no hormone testosterone in the female embryonic development, so that will cause the Wolfram duct degeneration. And then without DHT, the external genitalia will take on female characteristics versus those male characteristics. So the female embryonic development really is based upon the fact of having no SRY gene present. Without this being present, we will develop the female reproductive organs and will remove the Wolfram duct by degenerating it. In this next slide, we will look at a quick picture similar to the one from the male embryonic development, just discussing how the bipotential embryo will then come down and differentiate into the female. So here we can see at 10 weeks, the gonadal cortex becomes ovary in the absence of SRY protein. So again, there's no SRY gene in the female embryonic, and then under the influence of those specific female-specific genes. The absence of testosterone will cause the Wolfram duct to degenerate. So right here, we will not see that anymore. And then the absence of the AMH hormone will allow the Mullerian duct to become the fallopian tubes. So here we can see the process of the fallopian tubes being developed. And then we have the uterus, the vagina, and the ovaries. And then lastly, I just wanted to discuss some special considerations. So the presence of the Y chromosome is what determines if an individual will become a male. However, there are certain situations where where an individual or an embryo will have an abnormal chromosome linkage. So you can have an XXY, which is shown here, and it's noted that even just because there is 2 X's does not mean that it's a female. The presence of the Y chromosome determines that the individual will become a male. There's also a YO zygote. This is when there's no X chromosome in the zygote and actually results in the zygote dying because the larger X chromosome has essential genes on it that the Y chromosome does not, so it will not be able to survive. There's also something called an XO chromosome matchup where the XO will actually develop into a female. However, because there's not two X chromosomes, the normal reproductive systems for a female will not be able to function correctly. So without that second X chromosome, you will have an inactive reproductive system. That summarizes the determination of gender based on genes and hormones, and I hope you have a good night. Thank you.